My name is George Huber, and I'm going to give you the tour of two of the Huber labs. The first lab is 1023. In this lab, we have several reactors, analytical equipment, and characterization equipment. This is the Sitka system in a high-pressure olefin reactor. In this laboratory, we have 7 different gas chromatographs, 6 different batch reactors, high-pressure batch reactors you can see here. We also have a 2-dimensional. Our next laboratory is 1020 across the hall. In this laboratory, the first thing you'll see is a HPLC. We have 2 HPLCs in here. In this laboratory, we do some catalyst synthesis as well as several reactions. We have some high pressure batch reactors, and we have our catalyst preparation system. You'll see our reactors are mainly in little snorkel units to protect there. See a high temperature reactor system. And then down here, down in this hallway, you'll see our high pressure reactors. Our graduate students build these things, they operate them, and they do different reactor systems. We're studying conversion of biomass to different fuels, chemicals in this reactors. Here's 3 different reactor systems. All of these are hooked up to the different online gas GC systems that are right across from. Now we're going to walk across the hall to our Huber Group offices. And here's the Huber Group offices. We have shared office space. Each student get their own desk to store their equipment, store their books.
A lot of the plastics we use today for food packaging are actually made out of many different types of plastics. These are combined together to form a multi-layer plastic film. The different plastics in these structures provide specific properties like water and gas barriers, rigidity, and also plastic durability. Normally, these plastics cannot be recycled through traditional methods like mechanical recycling because they are very complex, and they usually just end in landfills. One way to increase recycling rates of these materials is to apply solvent systems to selectively recover each polymer component. And we're doing that as part of the efforts in Professor George Huber's group and the Chemical Upcycling of Waste Plastics Center at UW-Madison. In this new technology, we put small pieces of the multilayer film in a dissolution vessel, and we let the dissolution occur in the vessel for a given amount of time in a specific solvent. After dissolution is completed, we then separate the dissolved polymer from the rest of the polymer components. An anti-solvent is later added to precipitate the polymer component. At the end, a filtration step is done to recover the solid fraction of the polymer component. This way we have a new and efficient way of recovering and recycling the components in multi-layer plastic films. This process is known as STRAP, which stands for Solvent Targeted Recovery and Precipitation.
Hi everyone, my name is Jiayang Wu from Professor Huber's group. Today I'm going to talk about how to recycle PVC and why it is important to remove the chlorine from the PVC. Based on the plastics in European, around 51 million tons of the plastics are being consumed every year. According to this figure, PVC can be found on every categories of the plastic applications. However, the Environmental Protection Agency reported that no PVC products are being recycled in the US. What causes the PVC so hard to be recycled? This is because the hydrochloride will be generated when PVC gets heated up. More than half of the mass of the original PVC will convert to the hydrochloride, which is a very corrosive chemical. In this video, you can see when the 2 iron ions get into the hydrochloride solution, a severe reaction happens immediately. So if there is some hydrochloride in the feedstock of a recycling plastic stream, with years, the hydrochloride can eat through the reactor. So in the UW-Madison, our lab is trying to understand how to remove the chlorine from the PVC efficiently. The PVC powder will be put into a round bottom flask and heated up to 320 degrees C and hold for some time. During these reactions, the nitrogen will flow through the system and carry out any hydrochloride that is being formed, and then absorbed by some bases. The final product we get can remove 99.5% of the chlorine. and this product is ready to be sent to the process system. The pre-treated PVC will be mixed with other plastics together and sent into the left reactor, which is a process reactor, to convert those with plastics into the plastic oils. The plastic oils shown in bottom can be used to produce recycled plastic with virgin quality. And thank you. This is all for my today's presentation.
Hello, this is Minsu. Today I'm gonna talk about biodegradable plastics. We are living with plastic. The plastic production and consumption are growing, but the amount of recycled plastic is still low. A lot of plastic is discarded and incinerated because of usage for packaging. What is the biodegradation? Biodegradation is a kind of process that includes mineralization by microbial. From the degradation, there is the production of CO2, H2O, and CH4. The degradation of biodegradable plastics takes place in two steps. The first step is the degradation of the main chain of the polymer by microbial with hydrolytic enzymes and abiotic hydrolysis. The second step is the mineralization of the decomposed plastics during metabolism. The microbial consumes the decomposed plastic as their carbon source. Also, there are many factors affecting the biodegradation rates. There is a classification and examples of bioplastic. Bioplastic can be bio-based plastic and biodegradable plastic. Generally, biodegradation rate is a function of the polymer structure and does not depend on the origin of the raw materials. This means that the petroleum-based plastic can be the biodegradable plastic, whereas the bio-based plastic cannot be biodegradable. There are some misunderstandings about the biodegradable definition. PLA is commonly labeled as biodegradable, but will only be biodegradable in an industrial composting system. If a PLA were dumped into the ocean, it would take hundreds of years to break down. Biodegradation tests are conducted in soil, compost, freshwater, and ocean. According to the test, the biodegradation rates of each plastic are different. I would like to show you something with the biodegradation test in Hoover's research group.
This test is ASTM E5988. This test is to determine the rate and degree of biodegradation of plastic in soil at room temperature. This reactor includes the KOH and water, soil, and plastic in soil in the airtight desiccator.
This is the acid and base titration with HCl. I'm pouring the HCl to the KOH. At the pH change point, the color is changing rapidly. Biodegradable polymer can be used for many applications. However, it has difficulties in their low mechanical properties and their high cost. First of all, we must recycle plastic ourselves. Thanks for watching my video.